Hello, everyone. This is Dr. Karen's, and in this video, I'm going to talk about the materials and methods section of the Unknown Project Lab Report. So in this section, this is where you are telling the reader what experiments you did and what other things you had to use to identify your unknown. And I want this information presented in a very specific way. So one of the things you're going to include in this section is your dichotomous key, your complete dichotomous key, the one that you turned in to me and that I graded, and that because this is what you had to use to figure out what your unknown was. So this could be a flow chart or it could be the stepwise format, however you want to present it. Um, if it's a flow chart, it can be hand drawn, but it has to be neat, it has to be legible, and it has to be big enough for me to read, meaning it should take up probably the whole page or 2 pages if you need to use 2 pages, 1 page for gram positives and one page for gram negatives. That is fine. It has to be on the same paper, the same kind of paper that you're using for your report, and the page has to be numbered. So it's probably, and also remember, you are creating and uploading a digital copy of this paper, and you're handing in a paper copy of this paper. This dichotomous key has to be in both versions. So if you have to upload a digital copy somehow, you are going to have to digitize your key if you hand drew it. So it probably means if you're going to hand draw your flow chart, you're going to have to take a picture of it and scan it and insert that into your paper, but you have to make sure it's big enough that I can read it. If you're using the stepwise format, that can be typed directly within the document, no problem. Let's see. Oh, specify which set of data you used to make your key, meaning are you presenting the key that you originally turned in to me? If so, that means you created that key using the expected lab results or expected test results in our lab book. You have to include that information to the reader. Alternatively, maybe you rewrote your key after we did our practice round and we learned that some of our tests didn't behave as expected, and you used the abnormal class results to make a new key, which is totally fine if you want to do that, but you need to state that in this section. The second thing that's going to be included in this document is your materials and methods table. I want very specific headings on this table, and this should look very familiar. I want the name of the test that you did, the media and or the reagents that you used to conduct that test, what positive test results look like for that test, and what negative test results look like for that test. Please note that we are not talking about what results you got. We are talking about just in general. So for example, everyone's going to do a gram stain, so the gram stain should be in the materials and methods table. That will look like what? Test name, gram stain. Media and or reagents that were used, that's where you list all the different stains and mordants that you use during that staining process. And then appearance of positive results. Gram positive looks purple. Appearance of negative results, pink. That doesn't tell me anything yet about what you have, and that's fine because that's not what we're looking for yet. You're just you're giving the reader an overview of all of the tests that you conducted, a summary of all the tests that you conducted. When I said the headings on this table should look familiar, when we did the practice round, that was the table you had to fill out and turn in to me. I gave you a materials and methods table to fill out so I could give you feedback on it so that when you present it here, you're presenting the information that I'm looking for. That being said, do not just staple that key to your paper. Don't take a picture of that key and insert it into your paper. I want you to regenerate the key, and you need to do that within the document word processing program that you're using. If you're using Microsoft Word, you need to create the table in Microsoft Word. If you don't know how to do that, it's really easy. I can show everybody how to do that in class. Alternatively, you can also look that up online very quickly and find a YouTube video about that. If you want to create your table digitally in a different program and then insert that into your paper, you can do that as well. But it cannot be hand drawn. It cannot be a scanned image of the table that you turned into me. The materials and methods table should include only the tests that you actually conducted, including the gram stain. Now, some of you may decide that you're going to do all the tests. Fine, then all those tests should be in this table. Some of you may decide, well, I don't need to do all the tests because my gram stain told me information that eliminates some tests. That's fine too. Whatever tests you conduct for this whole experiment, they have to be in this table. Now, this section should not just be your dichotomous key and this table. You have to have a few introductory sentences at the beginning that describe to the reader what they're about to see. So tell the reader, what is the dichotomous key? What is it, and how did you construct it? The table. This is where you can reference the lab book. So I don't want you to describe in detail how you did each test. The information that I want is really just what you're going to present in this table. But you can add a sentence or two about where you got the procedures from. So you could say the following tests were conducted following the procedures in the lab book, and then you cite the lab book. But you need to have some introductory sentences. Now, this is what I don't want. Again, I said this earlier. Don't give me detailed descriptions of each test. I don't need you to rewrite the procedure for each test. That's ridiculous when you can just reference the lab book and then I can go look at that lab book and look at the procedures of that test. I don't want you to rewrite the procedures. I just want the information in the table. Don't be sloppy if you're going to hand draw your dichotomous key or if you're going to copy and paste images into this section. Make sure things are neat, legible, and big enough that I can read them. Take as many pages as you need to do that. The next section, I'm going to go through the results.